Very good evening to the students who have joined the class. We will just wait for 5 minutes and then we will start today's session. Meanwhile, the students who have joined the session, can you please just write in the chat box if my voice is audible and my screen is visible. Hello. Okay, thank you so much for the confirmation. We will just wait for a couple of minutes and then we will start today's session. So, is the screen visible? Can you write in the chat box if my presentation is visible to you? Okay, thank you so much. Just let's wait for a few minutes and then we will start today's session. Okay, let me introduce myself first. My name is Moshumi Dev and I am a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad, and I am the TA for your NPTEL course of Organic Chemistry in Biology and Drug Development. And today it is our week 7 live session where we will discuss the week 7 course contents, and it is based on the week 7 course content, which is consisting of 4 chapters, and we will discuss some 10 to 15 MCQs here which will help you to attend this week's assignment. And at the end of the session, if you have any doubt, please write in the chat box. We will wait for a few minutes and we will then start today's session. And you can use the chat box to know the course better, and if you have any doubts, so please write in the chat box. Okay, it's already 7 p.m. So let me just start our session. Again, I am going to introduce myself. My name is Moshumi Dev. I am a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad, and I am the TA for your NPTEL course of Organic Chemistry in Biology and Drug Development. Today is the week 7 live session for this course, and we will discuss some 10 to 15 questions today based on the week 7 course material, which is and this kind of question will help you to do your this week's assignment and to know the course better. And so I think we will just wait for 1 to 2 minutes more and we then start today's session. Let a few more people join and then we will start today's session. Meanwhile, if in the middle of the today's class, if at any time my voice is not clear or my video is not visible, please please write in the chat box, and if you have any doubt, any kind of doubt in middle of the session, please also you can use the chat box to address your query. At the end of the session I will answer those. I think since we have limited time, we should start today's session, and I will add up more students if they join meanwhile. Okay. So, the first question, it is said that which of the following statements is false regarding the restriction enzymes? Option A, these are endonucleases. Option B, these are exonucleases. Option C, these only recognizes palindromic sequences, and option D, it is saying that these digest double stranded DNA at specific sites. So, first I will show you the answer for this question, and then we will discuss about restriction enzyme. Again, this is a for true and false kind of question. So, please read the question carefully while doing this kind of problems, because these are this sometimes may be a little bit of confusing. So, they are student often made mistakes in this kind of question. So, please read the question carefully before answering. So, let me show you the answer first, and we will discuss about the restriction enzymes. Okay. So, the answer for this problem is number B, these are exonucleases. So, this is the false statement among this. Let me use a pen. So, this number B is the false statements among these four. Okay. So, let us discuss about the restriction enzyme, what they do and why the number B is the false. So, what is restriction endonuclease? So, restriction enzymes, they are often called restriction endonuclease also, because they actually cut the DNA. The restriction endonucleases as the family of the enzymes that each cut DNA at a specific sequence within the DNA. Alternatively, they are called restriction enzyme. A restriction endonuclease can cut the DNA when it recognizes a specific sequence. These specific sequences are called restriction sites. So, what does the restriction enzymes do? If you have an entire copy of DNA and you want to just replicate or clone a particular part of a DNA to study it further, so what you have to do, you have to cut the DNA at a specific age, specific phases. Okay. So, this is the work the restriction enzymes do. They are basically the cutter for the DNA. Okay. And they basically cut in the middle of a DNA. So, they are called endonucleases, not exonucleases, and they do not cut at the end. Okay. So, and the restriction endonucleases digest double stranded DNA at specific sites, and this recognition site is a palindrome syndrome. And what is a palindrome syndrome? It is actually the example is like Madam, I am Adam. So, if you say that the sequence, like there is M A D A M I M A D A M. Okay. Now, if you just reverse this word, okay, then we will have M A D A M, which is Madam, I M, which is I am, A D A M, Adam. So, if you read this line, it is the in forward and backward, both the sequence is same. Okay. Like if you have A G C T, and if you just have the complementary pair, which is T C G A, now it is the both the repeat, it is the same sequence at the forward and backward also. So, this is called a palindrome sequence. Now, some numbers are also called palindrome number, like if you write like 1, 2, 3, 2, 1. So, it is called also palindrome number, okay, because the sequence, if you go from here or if you go from there, you get the same sequence. So, these kind of sequences are called the palindrome sequence, and this restriction enzymes, they actually recognizes only the palindromic sequence, and based on these sequences there are several kind of restriction enzymes, like you have AIUL, ALUL, HAELL, BAMHI, HIND3 and ECORI. So, these are the some examples of these restriction enzymes. Somebody wants to join. So, these are the examples of the restriction enzymes. So, I think I am clear why the statement number B is the false regarding the restriction enzymes and what restriction enzymes are and what they do. And if you have any doubt, please write in the chat box. At the end of the session, I will address those doubts. So, I suppose this question and this answer is clear. Now, I will quickly move toward the next problem. Okay. So, the problem number 2, it says that for which restriction endonuclease the recognition site is bipartite and asymmetric, and the options are type 1, type 2, option B is type 2, option C is type 3 and option D is all of the 3. Now, what is the bipartite? The bipartite means there are two parts, okay. That is called bipartite. The meaning of the word bipartite, it means that it is consist of two parts. And asymmetric, you all know that the two parts, if it has two parts, they are not symmetric in nature, means they are not similar, they are different in nature. So, which of the following endonucleases is the recognition site is the bipartite and asymmetric? Let me show you the answer, then I will discuss the problem. Okay. So, the answer of this question is type 1. The type 1 restriction endonuclease, it has a recognition site which is bipartite and asymmetric. Okay. So, the restriction endonuclease which I discussed in the previous question, these are of 3 types, the type 1, type 2 and type 3. So, this is the chart which just compared the these 3 type of restriction endonucleases. Like for the structure, the type 1, it has a bifunctional enzyme which have with 3 subunits. Okay. The type 2, it consists of a separate endonuclease and a methylase. And the type 3, it also have a bifunctional enzyme just as a type 1, but it has 2 subunits, whereas the type 1 has 3 subunits. Now, for the recognition site, the type 1, it is bipartite and asymmetric. I again in the previous question I just what is the recognition site, that the recognition site is the specific sequence where the endonuclease is cut. So, for the type 1 endonucleases, the recognition site is bipartite and asymmetric. For the type 2, the recognition site, it consists of 4 to 6 base pair sequence, often palindromic. And again I discussed what is the palindromic sequence. If you go from forward and backward, it reads the same. Okay. And for the type 3, the recognition site, it has 5 to 7 base pair asymmetric sequence. Okay. Now, restriction and methylation. So, for the type 1, it is naturally exclusive. For the type 2, it is separate reaction, and type 3, it is simultaneous reaction. The cleavage site, for type 1, non-specific greater than 1000 base pair from recognition site, and from type 2, it is same as or the close to recognition site, and the type 3, it is 24 to 26 base pair downstream of recognition site. So, you do not have to remember all these things, you do not need to memorize. Just remember the recognition site, it is very important, because the for restriction in endonucleases, the recognition site is very important point. So, just remember this recognition site of this 3 type of restriction endonuclease. Okay. And if possible, please remember the structure also. Okay. So, I think it is clear. Then I will move toward the problem 3. Again I am saying please write in the chat box if you have any doubt regarding to any question, or at any point of the session, if you have my voice is not clear or the video is not visible, please write in the chat box. Okay. So, quickly I will move toward the next problem. So, now the problem number 3, it says that the palindrome sequence recognized by restriction enzyme hint 3 is number A, AAGCTT, number B, CTG, number C, GATATC, and number D, GAATTC. Okay. So, I explained in the first question that the restriction enzymes, they generally cut the DNA at the palindrome sequence. What the palindrome sequence is, if you go from forward to backward, it will read the same. As you can see, there is a AAGCTT. So, okay, first I will show you the answer and then I will discuss this. So, okay, the correct answer for this question is number A, AAGCTT. Okay. So, Now, if you why the sequence like this one? Okay, the hint 3 here, these are the some restriction enzyme, and I will suggest please remember few example of restriction enzyme, and the like there is a E. coli, this is a E. coli, like HA3, hint 3, PPUMI. There are several or restriction enzymes. So, please remember where this restriction enzymes cut, means the palindrome sequence recognized by the restriction enzyme. Okay, please remember this. So, why it is called palindrome? As you can see this example of hint 3, like if the DNA, you know that the DNA is double stranded, okay, and the first strand, if it is goes from 5 prime to 3 prime, the another one we will go it will go from 3 prime to 5 prime. As you can see that the if the sequence is AAGCTT, the in the next strand, okay, in the second strand, the sequence will be the base pairing, like adenine will pair with thymine and the guanine will be pairing with cytosine, and vice versa. So, the sequence will be TTCGA, and if you can see that if you read this sequence from this backward, okay, so it will read the TTCGA like this same. So, that is why it is called the palindrome syndrome, palindrome sequence, okay, because the forward and backward it reads the same. And the hint 3, it cuts between the two adenines of the this palindrome sequence AAGCTT. So, whenever in the DNA you will have this kind of sequence like AAGCTT, and if you use the restriction enzyme hint 3, then it will cut between the two adenines, okay. Now, if you have this restriction enzyme, suppose E. coli, this first one E. coli, then it will just recognize the sequence GAATTC, and it will cut between the guanine and adenine, the G and A, like in here, okay. And similarly in RV, it will cut between the T and A, like in between this. Now, T and A, it will cut in this place in the middle, and similarly in case of HAE3, it will cut between the G and C, and in hint 3 it cut between A and A, and in this PPU MI, it will cut between the G and G. And what this G and W, so why this means, it is written here that the abbreviation R, it represents the purine, adenine and guanine, and the pyrimidine, cytosine, thymine and uracil, it is abbreviated as Y, and the abbreviation W, it represents the adenine and thymine, okay. So, I think the problem, this problem 3 is clear to all of you. So, please I will suggest remember few restriction enzymes and the palindromic sequence recognized by these. This is very important topic for your examination purpose also. So, please remember, okay. Now, I will quickly move toward our fourth problem. Okay. So, the number 4, it says that which of the following statements is false regarding the plasmid PBr322? The option A is PBr322 plasmid contains 2 genes. Option B, it is a natural plasmid. Option C, one gene confers resistance to ampicillin, and option D is other gene confers resistance to tetracycline. Okay, so PBr322, it is a plasmid, it is basically a vector which is used for DNA cloning, and I will show you the answer for this question, and then we will discuss this plasmid PBr322. Okay, the correct answer is it is a natural plasmid, okay. So, it is the false statement actually. So, PBr322 is not a natural plasmid. So, the plasmid actually it contains a circular DNA, okay. So, I will show you okay. So, yes. So, as in this figure, if you can see here, I will just point the laser pointer. Okay, as you can see, it is a plasmid of PBr322. This is a circular DNA, and it consists of 2 genes, and one gene, this is in the green color, it is resistant to the tetracycline. Here it is written TET, TET, and another gene which is at the, this is marked as this red part, it is resistant to the ampicillin, okay. So, what is the PBr322? PBr322, it is not a natural plasmid, it is actually a purpose built plasmid vector and was one of the first widely used cloning vector. It has a relatively small size of 4363 base pair, This is important because transformation efficiency is inversely proportional to size, and above 10 kbp is very low. The nomenclature of PBR 322. So, it is created in 1977 in the laboratory of Herbert Boyer at the University of California, San Francisco. The P, the word letter P, it indicates as a plasmid. The BR, it identifies Bolivo and Rodriguez, the two researchers who developed it. 322 is distinguishes those plasmid from others like PBR 325, PBR 327 developed in the same laboratory. So, PBR 322 is a 4363 roughly base pairs in length and has two antibiotic resistant gene. The gene BLA encoding ampicillin resistance protein, this is the part, this red one, and the gene TET-A, it encoding the tetracycline resistant protein, this green one. Okay, and it contains the origin of replication of PMB1 and the ROP gene, which encodes the register of plasmid copy number. The plasmid has a unique restriction size for more than 40 restriction enzymes. So, this plasmid DNA, there are more than 40 recognition site for the 40 more than 40 restriction enzymes like the E. coli, the Hint3. Okay. The 11 of this 40 sites lies within the TETR gene, means this green part. Here more than there are 11 of this restriction site lies. There are two sites for the restriction enzyme Hint3 and CLA1 within the promoter of the TETR gene. Means at the promoter region there are the restriction site of two restriction enzyme Hint3 and CLA1, and there are 6 key restriction sites inside the AMP gene. Okay. So, this is the some information about this plasmid PBR 322, and I think it is the enough information for this plasmid. So, please remember, like, it is not a natural plasmid, it is a laboratory made plasmid, and it contains mainly two gene, and this two gene, one is for resistant to the ampicillin, one is encodes the protein which is resistant to the tetracycline, which is antibiotic, and another also is a antibiotic resistant protein encoded gene, which is for the ampicillin antibiotic. Okay. And there are more than 40 recognition site within this cyclic DNA of this plasmid for the 40 restriction enzymes, and the like Hint3 and E. coli, and also remember, like for this Hint3 and the CLA1, they lies in the promoter region of TTR gene, which is the which encodes the protein for the tetracycline resistant. Okay. So, remember this thing about the PBR 322. It is also very important topic for your course. So, please read it carefully, and if you have any doubt, please write in the chat box, or at the end of the session I will address those doubts. So, let me quickly move toward the next question. Okay. The problem 5, it is said that the first step in PCR technique deals with number A, binding of primers to a single stranded DNA, option B, chain elongation by DNA polymerase, option C, denaturation of DNA, and option D, none of this. So, I will quickly show you the answer first, then we will discuss the steps in the PCR technique. So, the option is the correct option is C, the denaturation of DNA. So, it is the first step in the PCR technique. Okay. Now, as this is a beautiful flow chart given for the various steps in the PCR technique. The PCR stand for the polymeric chain reaction, and it is a technique for the cloning of the DNA. Okay. So, the step 1 for the PCR is the denaturing. As you can see, I will just quickly on the laser pointer. Okay. The step 1 is the denaturing, and it just look at the temperature, it is the 95 degree centigrade. So, at this centigrade, the two strands of DNA, the denaturation is happening. Okay, and we get from this double helix structure, we are getting the two DNA template strand. One goes from 5 prime to 3 prime, and another one from 3 prime to 5 prime. And now, the step 2, it is the aniline. Okay. In the annealing step, we use the DNA primers. Okay, in the orange and green, you can see there are few DNA primers, and from one primer it is binding at the goes from 5 prime to 3 prime end and bind at the 3 prime end of this first strand, and another one it goes from the 5 prime to 3 prime end is another binds in the 3 prime. So, the DNA primers it generally bind to the 3 prime end for the strands of the DNA. Okay. So, and now we have some nucleotides. Okay, here you can see in this figure there are nucleotides, and there is a very important enzyme DNA polymerase TAQ, tuck polymerase. Okay, and this is a heat stable polymerase, DNA polymerase. As you know that the DNA, the polymerase, it is basically a enzyme, and enzymes are nothing but the proteins, and at the high temperature the protein that generally get denatured. Okay. So, here you can see the temperature is 95 55 degree centigrade, when the human body temperature it is 37. So, basically the enzymes work in roughly at that 37 to 40 degree centigrade maximum, but at the 55 95 degree centigrade they basically this enzyme they will be denatured and this will not work. But this tuck polymerase it is actually a heat stable polymerase, okay, which is used, and there are also some very few heat stable polymerase which are found in the hot springs and in this kind of places, and there are also cryopolymerases or cryo enzymes which are found in very cold temperature. So, these are some special kind of enzymes. So, what this the tuck polymerase do, it basically joins the primer this nucleotides with the primer, and the new strands of the DNA are formed. Okay. So, it is the step 3, it is the synthesizing which happens in the 72 degree centigrade, and after this synthesizing the cycles are repeated to the 20 to 40 times to get the desirable clone of DNA, and you can see that from 2 DNA strands we are getting 4. Okay. So, the normal common formula for this polymer is it is the 2 to the power n. Okay. So, after the if you are asked that after 2 cycles what is the number of the DNA, so total number of DNA, so it will be 2 to the power 2, it will be 4. Okay. So, for this DNA PCR technique, please remember the steps. The first step is the denaturing, the second step is the annealing, and the third step is the synthesizing, and remember the temperatures also. Like for the denaturing step the temperature is 95 degree centigrade. For the step 2 annealing step the temperature is 55 degree centigrade, and for the third step the temperature is 72 degree centigrade, and the enzyme is used, the DNA polymerase used is the tuck polymerase which is a heat stable polymerase, and we are using DNA primers which binds from the 3 prime end and goes for 3 prime end in the both strands, and the nucleotides are there are some nucleotides which are joined with this primer by this tuck polymerase, and we get the new strand of DNA. Okay. So, this is all about the PCR technique, and I hope it is clear, and I will move toward the next problem. Okay. Okay. So, this is the problem number 6 which is said that suppose you are doing a PCR to amplify the portion as indicated in the red on the following piece of DNA. So, how many copies of the amplified portion of double strand DNA you will obtain for after the 10 cycles? The options are 2 to the power 10, option B is 2 to the power 8, option C is 2 to the power 6, and option D it is 2 to the power 9. And now this is a very interesting question, because in the previous slide I had said that generally the number will be 2 to the power n. So, generally it should be 2 to the power 10, but the correct answer for this question it is 2 to the power 8. Now, I will tell you why. So, as you can see this is the entire strand of this DNA, but we do not need the copy for the whole DNA strand, as the question says that you are doing a PCR to amplify the portion indicated in red. So, we only need the copy for this region, okay, for this region. We do not need this region. So, at first when we will use the primer, the DNA primer will bind to this 3 prime end of the both strand, and it will start the cycle. Okay. So, after first 2 cycle we will get a copy which we do not require. Okay. So, after seeing that we do not require, we will start melting this DNA strands, and after the melting the primer, now when it will have this sequence, it will bind to this end, and we will get the our required portion. Okay. So, generally the first two cycle we are do not having any our required in the first two cycle after the PCR we are not getting our required portion, the amplification of our required portion, since it is in the middle of the DNA strands. Okay. So, remember, if it is a normal PCR, if you are getting a normal strand and you are doing the normal PCR, the number will be 2 to the power 10, but if you are asked to how many copies you will get from this kind of marked region which is in the middle of this DNA strands, so it will always be the 2 to the power n minus 2. 2 to the power n minus 2, because the first two cycles these are wasted for this extra regions which we do not require. Okay. So, the number is 2 to the power n by 2 for the this kind of sequence where there is a marked region, this red one. Okay, the marking is not proper. So, this is the formula you need to remember. So, it is a very easy, just remember, if it is normal then the formula is 2 to the power n, but if there is a this is indicated region in the middle, then it will the formula will be 2 to the power n minus 2. Okay. Now, I will move toward the next problem. Okay. So, see, the PCR technique, it is a very important for your course. So, please read this technique carefully, and the for next problem it is also regarding the PCR. So, it is saying that which of the following statements is false regarding the PCR primers. Okay. So, the options are: number A, DNA polymerase requires primers to initiate replication. Option B, PCR primers are short and single stranded. Option C, primers are sequence specific and bind to particular genome sequence, and option D, higher the length of the primer, lesser will be the selectivity. So, we need to identify the false statement regarding the PCR primer among these four, and I will show you the answer, then we will discuss the PCR primers. So, the incorrect statement among these four are: number D, higher the length of the primers, lesser will be the selectivity. So, the primers are very important part for the PCR technique, and there are some consideration about these primers, like the characteristic of the primers, the specificity, the specific for the intended target sequence, and the thoughts on the these are some thoughts for the primer design. As you can see, let me show you the laser pointer. Okay. So, these are some thoughts for the primer design, like the specificity, the uniqueness, length, base composition and internal stability. Okay. For the stability, the melting temperature, annealing temperature, internal structure, and the compatibility, the primer pair matching. Okay. So, general rules for a primer design, the primer and amplicon length. The primer length determines the specificity and the significantly affect the annealing to the template. The too short means low specificity resulting in non-specific amplification. Too long decrease the template binding efficiency at the normal annealing temperature due to the higher probability of forming secondary structures such as hairpins. So, optimum primer length, it should be 18 to 24 base pair for general application and 30 to 35 base pair for multiplex PCR. And optimal amplicon size, the 300 to 1000 base pair for general application. So, avoid greater than 3 kb and 50 to 150 base pair for real time PCR, and avoid greater than 400 base pair. Okay. So, these are few general rules for the primer design, and why these other statements are correct. So, all we know that DNA polymerase this requires the primers for the initiate the replication, and PCR primers are short and single stranded. Primers are sequence specific and bind to particular genome sequence, and the higher if the length is short, then the specificity will be lower. So, that is why the number D, it is the false statement among these four. Okay. Let me just check if it is there is something in the chat box. Okay. So, I will quickly move toward our next problem. So, I hope it is clear, and if you have any doubt, any problem, please write in the chat box. Okay. At the end of the session, I will just discuss those doubts. Okay. The problem number 8, it says that in Huntington disease, the trinucleotide sequence that repeats more than 36 times is: number A, CAA, number B, AGC, number C, CAG, and number D, TAC. So, I will share this Huntington disease is a genetic disease, okay, and this is due to the the abnormal gene coding, and so I will show you the answer. The answer is number C. So, I will show you. Okay. So, there is a in the Huntington this trinucleotide sequence CAG, if it is repeats more than 36 times, so then we will have this abnormality in the gene, and this disease will be formed. Normally it is less than 36, and if it is less than 36, then there will not be any kind of problem, but if it is greater than 36, then this disease will be happening, and this is the technique how this is identified. I mean, the blood sample is taken, and the PCR is done, and if it is Sanger sequencing and long read sequences, it is done, and it is identified by this techniques, and for the normal gene it is less than 36, but for the for this disease happening, it is greater than equal to 36. And the clinical features for this Huntington disease, it is the motor, the cognitive, and the psychiatric. So, the motor this is features is the chorea, the dystonia, motor impersistence, dysarthria, dysphagia, poor posture, in coordination, impaired gut and balance, delayed initiation of voluntary saccades. And the cognitive, it is the loss of the excessive executive functions, difficulty in multitasking, rigid and inflexible thoughts, poor concentration and easily distracted, impaired spatial perception, impulse control and disinhibition, depreciation of vocabulary, poor insight, and short term memory loss. And the psychiatric features are depression, anhedonia or decreased libido, suicidal ideation, social isolation, disrupted sleep pattern, delusions, hallucinations, paranoia or irritability. Okay. So, you don't need to remember all these features, it is just for your understanding. And so, remember this only remember the sequence, like the CAG, it repeats more than 36 times for this Huntington disease, and for normal cases it is less than 36 times. Okay. So, just remember these two things and it is enough. So, next I will move toward the problem number 9, and it says that which of the following statements is false regarding the cystic fibrosis. So, cystic fibrosis is another gene induced disease, I should say, and the options are: it is a genetic disease. Option B, PCR can detect this disease. Option C, in non-CF individual, the CF means the cystic fibrosis individual, CTFR gene codes for the protein, that is sodium ion channel. Okay. And option D, in cystic fibrosis individual, mutation in CTFR gene leads to the thick mucus secretion in the lungs. Okay. So, the disease of cystic fibrosis, these are the 4 statements about this disease, and you have to identify the false among these 4. I will just first show you the answer, then we will discuss this disease and why the answer, which is the following correct answer. So, the incorrect statement for this question is the number C. In non-CF individual, CTFR gene codes for the protein, that is sodium ion channel. So, it is the cystic fibrosis, it is a genetic disease. Okay. The gene mutation in this is the reason for this kind of disease, and PCR can disease this gene induced disease. Okay. So, this in the CF individual, the mutation in CTFR gene leads to the thick mucus secretion in the lungs. Okay. So, as you can see, this is a diagram. Okay. So, number C is the incorrect statement. Okay. In the non-CF individual, the CTFR genes does not code for the sodium ion channel, but for the chloride ion channel. So, as you can see that there is a this is the cell wall, and the CFTR protein, it is in middle in this cell wall, it is and the chloride ion can pass freely for this protein. Okay. So, when the mutation happens, the CF lung cell, the problem one is function. Okay. The chloride ions can reach the channel at the cell surface, but cannot pass through, and this has been called the Doorman problem. The mutant CF protein needs a correct corrector drug to boost it to the surface. It is also needs a Doorman drug to open the channel so the chloride ion can pass through. So, basically in this genetic mutation what happens that this this is the door, this is a door for the passage of the chloride ion, and the chloride ion can pass the this is the lung cell. Okay, the chloride ion can pass easily from this CTFR protein, but when the mutation happens this is basically closed and the chloride ions are trapped between the cells, and because of that a thick sticky mucus buildup happens which flattens the cilia of the cell wall. Okay, and for the normal DNA the CFTR protein develops normally and reaches the cell surface and becomes an open channel, the door for the chloride ions. The door jamming mutation including G551D, this mutation affecting LoRa and the Kate Chivers disable function at the cell surface, and common delta F508 mutation, the CFTR protein is made, but it is just floats around the inside the cell without reaching the surface. Okay. So, there are two kind of mutation for because of the cystic fibrosis can happen, and it happens in the CTFR gene, and it is basically for the coats the protein for the passage of the chloride ion. So, just remember this 3 to 4 things like why the gene for the cystic fibrosis, because of that cystic fibrosis happens, it is the CTFR gene and it codes for a protein for the chloride ion. And there can be two kind of mutation. One mutation the door is jammed for this chloride ion passage, and for another the protein is not made, but it does not stay in the surface. Okay. So, remembering this few is the enough for the cystic fibrosis. So, I will move toward the next problem. Okay. So, the problem number 10, it says that identify the incorrect statement among the following. Number A, coenzymes are either small organic molecules or inorganic metal ions. Number B, apoenzymes are activated by cofactor. Option C, prosthetic groups are coenzyme which binds permanently with the enzyme. And option D, cofactor is a non-protein molecule. Now, you have to identify the incorrect statement among the following, and let me tell you this coenzyme, cofactor, prosthetic group, apoenzymes, these terms are very much confusing confusing. Okay, I mean I myself get confused several times for this kind of terms. So, you just need to just focus on a flow chart kind of thing, so that you can remember this thing. Okay. Okay. The correct answer is the number A, coenzymes are either small organic molecule or inorganic metal ion. So, let me discuss what is the coenzyme, what is the apoenzyme, prosthetic group, cofactor. Let us see. So, okay. So, cofactors, the general terms are the cofactor. Okay. Let me use this laser pointer. So, the general term is the cofactors. So, what are the cofactors? Generally enzymes, some few enzymes they are not activated as it is. Okay. So, the inactivated form of the enzymes are called the apoenzymes. Now, the cofactors are a extra part which is this extra part, it is the non-protein. Basically the cofactors, these are the non-protein extra part which upon joining to the apoenzymes make activated form of enzyme which is called the holoenzyme. So, there are several part of the cofactor. The first one is the essential ions and the second is coenzymes. Now, the essential ion, these are the inorganic ions. Okay, and they can be two parts. Okay, first is the activated ions which are the loosely bound, and the metal ions of metalloenzymes which are tightly bound. So, metalloenzymes, you know that the enzyme which has a metal center and it is tightly bound to the enzyme, and it is very important for the activity of the enzyme. And there can be activated ion which are the loosely bound, means they are not just tightly bound to the enzymes, and these are the inorganic part. And the cofactors can also be organic. Okay, organic molecules, and if it is organic it is called the coenzymes. Okay. So, this coenzymes are of two, it can be type 2. First one, it is called co-substrate, and this co-substrate, it is loosely bound with the enzyme, and prosthetic group which is the tightly bound with the enzymes. Okay. Okay, somebody I think wants to join. So, where I was? So, the coenzymes are the organic molecules and they can be of two types, co-substrate and the prosthetic group. Okay. So, now you understand why the number A is incorrect, because it is saying that the coenzymes are either small organic molecule or inorganic metal ion. So, it is not correct. The coenzymes are small organic molecules. If it is the cofactor which can be either the organic molecule or inorganic metal ions. Okay. And the coenzymes are of two types, co-substrate and prosthetic group. The co-substrate is loosely bound and the prosthetic group is tightly bound. And the coenzymes function as a transient carrier of specific functional groups. Okay. So, it is saying it is written here also that a large number of enzymes require an additional non-protein component to carry out its catalyte function, and they are called as cofactors. A coenzyme or metal ion is covalently bound to the enzyme protein is called the prosthetic group. The cofactors are two types, the inorganic ions such as ferric and Fe2 plus, magnesium, manganese, zinc, and number 2 is the complex organic molecule called coenzyme, and some enzyme require both a coenzyme and a metal ion for their activity. So, just remember this flow chart, and it is very easy if you remember this flow chart, and otherwise you will get confused what is the cofactor, what is the coenzyme, what is co-substrate. Okay. So, remember this flow chart and it will be easier. So, quickly I will move toward our next problem. So, okay, the number 11, problem number 11, it says that which lipid soluble vitamins act as a precursor of coenzymes. Number A, vitamin A and D, number B, vitamin A and K, number C, vitamin E and K, and number D, only vitamin E. So, as you know that vitamins can be of two types, it can be lipid soluble or fat soluble, and also it can be water soluble, and the vitamins A, it is called ADEK. Okay, A D E K. So, these four enzymes, they are the fat soluble enzyme, and other like vitamin B, vitamin C, so etc. So, these are the water soluble enzyme. Okay, these are for the water, and this is the lipid soluble, this lipid soluble, these four are the lipid soluble. Okay. So, among these four, which vitamins act as a precursor of the coenzymes? The correct answer will be vitamin A and vitamin E. Okay. Okay, I am very sorry, there is a typographical error here. I will just correct it. Okay, the option number B is correct, so it will be option A, vitamin A and vitamin K. Okay. So, vitamin A and vitamin K, they are the precursor for the coenzymes, and these are called the pro-coenzymes. So, there is a chart comparing these four vitamins, vitamin A, D, E and K. These are the fat soluble vitamins, and the purpose of these four vitamins: for the vitamin A, it is the vision and the growth, development, immune function. For option D, for vitamin D, it is the bone growth and the development in children, bone maintenance in adults, and blood calcium regulation, insulin formation and secretion. For vitamin E, it is the antioxidant, and option K, for vitamin K, it is the blood clotting and bone formation. And the deficiency, what the deficiency of these vitamins are lead to? See, the vitamin A deficiency, it leads to the night blindness, hardened scaly skin, reproductive issues and increased infections. Option for vitamin D, it is the rickets, osteomalacia and osteoporosis. For vitamin E, it is the rare from diet, because vitamin E, which is very rare deficiency happens, and the fat malabsorption syndromes and the preterm infant. And vitamin K, it is rare in adults also, it is the fat malabsorption syndromes and prolonged antibiotic use. Okay. The toxicity, these are the some toxicity for this vitamins, and the sources from where you can have these vitamins. You all know that the fruit, the yellow, orange vegetables and leafy green vegetables, it is a good source of the vitamin A, especially the carrots and also the yellowish orangish fruits. And for the vitamin D, the sunlight, the early morning sunlight is a very good source for the vitamin D, and the food, for the food, it is coming from the milk and cereals. Option 8 is basically the wheat germ oil, this kind of seed oils. These essential oils are very enriched for the vitamin E. And vitamin K from plant fruits, soybean, canola, these are the very good source for the vitamin K. So, remember the vitamin A and K, these are act as a precursor of the coenzyme and these are called the pro-coenzymes. Okay. Now, problem number D, it is a very important question. So, it says that which of the following reactions take place in the vitamin D3. So, the correct answer for this, the options are, okay, I will just show you first the answer questions. So, the options are the option A, disrotatory ring opening followed by 1,5 hydrogen shift, conrotatory ring closing followed by 1,5 hydrogen shift. Option C is disrotatory ring closing followed by 1,7 hydrogen shift, and option D, conrotatory ring opening followed by 1,7 hydrogen shift. I will first show you the answer for this question, then we will discuss what is this conrotatory, disrotatory and this hydrogen shift, what does it mean. So, the correct answer is the number D, conrotatory ring opening followed by the 1,7 hydrogen shift. So, basically this conrotatory, disrotatory ring opening, ring closing and this hydrogen shift, it is a part of a pericyclic reactions. So, what is a pericyclic reaction? The reaction which goes in the cycle. So, basically as you can see in the figure, like this reaction, the electrons are moving in a circular motion, okay. That is why it is called the pericyclic reaction. So, there are several kind of pericyclic reaction, like it is the actually this ring closing, ring opening, it is the electrocyclic reactions, and there are also sigmatropic reaction, there are also which is called the Diels-Alder kind of reactions, there are also this kind of reaction, okay. So, what happens in this ring closing and what is conrotatory and disrotatory. So, actually in the it is called the 6 pi electrocyclic because there are 6 pi electron. As you can see in this, let us see in this middle structure, it has 3 pi bonds, okay. So, this 3 pi bonds, it contains the 6 pi electrons, and this 6 pi electrons actually moved in the circular motion, like if there is a, I will draw it here. So, as you can see the structure. So, here is a double bond here, here is a double bond and here is a double bond, and there is some groups here. So, there are 3 pi bonds and 6 pi electron, and if it is moving in the circular motion like this, so what you will have? You will have a closed structure and you will have this kind of structure. So, it is called 6 pi because 6 pi electrons are moving in a circle and we are getting this product from this, and the conrotatory, if the rotation these are in the clockwise direction, then it the electron movement, it will be said the conrotatory, and if it is in the anti-clockwise direction, it will be called disrotatory, okay. So, for the 6 pi system, the photochemical condition, it gives the 6 pi electrocyte in photochemical reaction, the it is it gives conrotatory ring closing, and in the thermal condition it gives the disrotatory ring closing or ring opening, okay. So, what is the hydrogen shift? Now, in this figure you can see that there is a hydrogen here, and if the hydrogen shifts from this side to this side, it is the 1,5 hydrogen shift. Now, how this nomenclature? The bond, you can see that this bond is breaking here. From this starting to this product, this bond is breaking. So, we will mark one here and one here, okay. Now, if you get this 1, 2, 3, 4 and 5. So, this hydrogen is shifting from this one position to this fifth position, that is why it is called the 1,5 hydrogen shift. Similarly, from the parent to product, this is 1 and this is 1 also, because this bond is breaking. So, if you number, give the number to this carbons, so it will be 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, it is 5, 6 and 7. So, this hydrogen is transferring from first to the 7th carbon. So, it is called the 1,7 hydrogen shift. Now, very interestingly, let us come to the vitamin D3, and it is a very good example of a pericyclic reaction in the in this our human body. So, we in the body we have 7 dehydrocholesterol, okay, and the structure is like this. So, why it is called that in the being in the sunlight we will have the vitamin D. So, it is because of this pericyclic reaction. So, basically sunlight means it is the photochemical condition, the H nu condition. So what will happen in this photochemical condition? First you will have a 6 pi conrotatory ring opening like this in this ring. Okay, you will have a 6 pi conrotatory ring opening, and we will have this P vitamin D3 by this first pericyclic reaction. Now in the second pericyclic reaction, the 1,7 hydrogen shift, and now again 1,7 hydrogen shift, it is favorable in the H nu condition, means in the sunlight, and 1,5 it is in the, actually it is in the thermal condition favor, and it is the H nu condition favor. Why it is favor, why it is disfavored? There is a very fast study for that, and you do not need to know everything about it. So just remember this 1,7 hydrogen shift happens. Okay, this hydrogen, there is a, if you can write it is a CH2, and you can have this hydrogen here. So it is 1, 1, 2, 3, 4, 5, 6 and 7. Okay, 1, 2, it is 3, it is 4, it is 5, it is 6. Okay, the numbering, there is a something, let me just correct it. Okay, so if you give the numbers, sorry, okay, so if you just give the number here, this carbon, it will be 1, and it will be 1, it will be 2, 1, 1, 2, it will be 3, it will be 4, it will be 1, 2, okay, it will be 2, 3, 1, 2, 3, 4, 5, 6, it will be 7, okay, 5, sorry, and it will be 4, 3, it will be 2, and it will be 1, because here it is starting from this bond, there is a hydrogen, and it will be 1, and it will be 1, it will be 2, 3, it will be 4, 5, 6, 7, and it is a 1,7 hydrogen shift will give us this calciferol, which is the vitamin D3, this called calciferol. So just remember that in case of vitamin D3, there are two pericyclic reaction, which is a conrotatory ring opening reaction followed by a 1,7 hydrogen shift, and it is these are called pericyclic reaction, and it takes place in our own body, and that is why it is happens in the sunlight, and that is why it is called that being in sunlight is good for the vitamin D formation in your body. Okay. So it is a little bit of difficult to understand for those who are not for the chemistry background. So I suggest to read some chemistry core chemistry book for understanding this pericyclic chemistry. Okay. So I will suggest at the end of the session, I will suggest some organic chemistry books for this understanding this pericyclic reaction also. Okay. So now I will move toward the next problem. So now problem number 13, it says that oxidation of retinol to retinal is catalyzed by option A retinal dehydrogenase, option B retinal reductase, option C retinal isomerase, and option D retinal deoxygenase. So I will show you the answer. The correct answer for this problem is option number A, the retinal dehydrogenase. And all you know that retinol, it is for the vitamin A metabolism. So this is the all trans retinal ester, and the first enzyme is the ester hydrolase. We will just hydrolyze this ester and make it this alcohol part, and it is all trans retinol, it is the vitamin A. Okay. Now then we will have this retinol dehydrogenase enzyme, and it will just oxidize this alcohol to the aldehyde, and it is the all trans retinal. So what is the role of this enzyme retinal dehydrogenase? So basically it is doing the oxidation of this alcohol to this aldehyde, and this all means alcohol, and this al means al suggests aldehyde. So the retinol to retinal transition, it is catalyzed by the enzyme retinal dehydrogenase. Okay, dehydrogenase itself means for the oxidation. Okay, mean the dehydrogenase means it will just remove two hydrogen molecules, and we will have this from alcohol, this aldehyde, and this all trans retinal, it is again an enzyme, it is called aldehyde dehydrogenase. It will make it the retinoic acid. So this question I have given like retinol to retinal oxidation. Also the question can be given to retinal to retinoic acid oxidation, and the enzyme will be aldehyde dehydrogenase. So, remember these two names for this two enzymes. The first one, the retinal dehydrogenase, which transforms retinol to retinal, and aldehyde dehydrogenase, which transforms retinal to retinoic acid. So, since we have very less time remaining, I will just quickly move to the next problem. Okay, the problem number 14, it says that identify the correct statement among the following. Option A, in PLP the 3OH is needed to be protected by phosphate linkage. Option B, aldehyde at 4 position will not show full activity if 5 hydroxyl methyl is not protected. Option C, PLP dependent racemase will convert D amino acid to L amino acid. And option D is PLP is not required for the decarboxylation of amino acids. So, I will show you the answer first, then we will discuss. The correct answer for this problem is option B, aldehyde at 4 position will not show full activity if 5 hydroxyl methyl is not protected. So, what is PLP? PLP is the pyridoxal phosphate, and it is derived from vitamin B6 family of vitamins. Okay. PLP is a coenzyme for enzyme catalyzing reactions involving amino acid metabolism such as isomerization, decarboxylation and transamination. So, these three reactions involving amino acids is catalyzed by this PLP coenzyme, and you can see that it is the structure of this PLP. Okay, this is the structure, and there are three part before going to this PLP. These are the first is called pyridoxine, and basically here you have 3 OH and is 5 OH, and this 5 OH, it is needed to be protected by this 5 hydroxyl methyl is needed to be protected by phosphate. Otherwise this aldehyde, this 4 aldehyde, it will not show its full efficiency, because if you have a aldehyde at this position and alcohol in this position, free alcohol, it will form a hemiacetal formation. Okay. So, if it is there is a hemiacetal formation between these two functional groups, the activity of PLP will not be full. Okay. So, because of that, this 5 prime OH group, it needs to be protected, and it is protected by this phosphate linkage. Okay. And other options are incorrect, because it is required for the isomerization, decarboxylation, transamination. So, quick just you need to, for this PLP you need to remember this the reaction, it is catalyzed like isomerization, decarboxylation and the transamination reactions, and there is the 4 prime OH, it is oxidized to the aldehyde, and the 5 prime OH, it is needed to be protected by the phosphate, otherwise this aldehyde will not show the full activity, and this aldehyde activity is very crucial for this PLP coenzyme activity. Okay. Now, I will quickly go to the last question for today's session, and the problem number 15, it says that which heterocyclic rings are present in the TPP. TPP stands for thiamine pyrophosphate. It is also a coenzyme just like the PLP, and this is the options are the pyrazole and indole. Option B is the pyridazine and oxazole. Option C is pyrazine and quinaxolone, and option D is pyrimidine and thiazole, and the correct answer is the option D, pyrimidine and thiazole. Okay. So, I will quickly go to the answer. Okay. So, what does this TPP, how it is formed? The thiamine, we know vitamin B1, it is called the thiamine, and the ATP goes to the AMP, and TPP, by the help of the TPP transferase, it is converted to the thiamine pyrophosphate, and this thiamine pyrophosphate, this is the structure, and there are 3 parts of this structure. The first one is the substituted pyrimidine ring, it is called the pyrimidine. The 6-member ring having nitrogen at the 1 and 3 position, it is called the pyrimidine, and then we have a substituted thiazole, a 5-member ring having sulfur and nitrogen at 1 and 3 position, it is called the thiazole, and we have a pyrophosphate ring. This pyrophosphate comes from the ATP, and ATP is converted to the AMP, and this substituted pyrimidine and thiazole, they are actually the thiamine, and this after joining with the pyrophosphate, it is forming TPP, thiamine pyrophosphate. So, for this kind of question, please remember the name of the few heterocyclic cores which are vastly used in the biological studies, like the imidazole, pyrazole, isothiazole, thiazole. These are the few heterocyclic cores, and it is very important to remember the names for this heterocyclic cores along with their structures. And further in this course, when the drug discovery chapter will be starting, when it will start, then you can see that there are very vast use of this kind of heterocyclic course. So, I will suggest you beforehand remember the just know the structure for the few heterocyclic score. Okay. So, that is it, that is for all for today. So, okay, if you have any doubt, any question, please, today's session is time is over. So, I will quickly share my email ID. If you have any doubt, you can mail me, or you can write first. Okay. So, this is my email ID. If you have any query about today's session, please write in the chat box. I will answer those query. Write in the email to me, because today's time is over. So, if you have any doubt regarding today's session, please write a mail, or you can write in the discussion forum. So, today it was the week 7 live session for this course of organic chemistry or biology and drug development. This PPT and today's class lecture video, it will be uploaded to the NPTEL website. You can download it from there and you can read it. So, thank you for today's session. I will now just close the meeting for everybody. So, thank you for joining. I will be closing today's session. Okay. Thank you so much.